Scientists believe there is a virus like Flogan involved from the Tenebaclum family or Dysnovirus that is causing sea-star wasting syndrome. In addition, natural causes such as water with warmer temperature may also be contributed to SSWS. Also, a lack of balance in microbes or sea-star may increase the progression or the disease. Some of the causes for SSWS are lesions found in the ectoderm causing the tissue to deteriorate. This leads to the decomposition of the sea star and its death. Also, a sea star shrinking display can occur before other symptoms or sea star wasting syndrome. 11 different species or sea star are affected by sea star wasting syndrome. These species are sunflower stars, rainbow stars, giant pink stars, giant mottled stars, ochre star, sun stars, letter star, vermilion star, six-armed stars, and bat stars.
Sea star wasting syndrome is mainly on the west coast because people have noticed that there are fewer sea stars in the intertidal zones in this area. And as early as 2011 and by 2015, most sea stars in Central Basin, Hood Canal, Northern Straits, Outer Coast, South Puget Sound, and Strait of Georgia, and the San Juan Islands have been affected by sea star wasting syndrome. Many of the sea stars had died off almost to the point of extinction. It is unknown if sea star wasting syndrome can affect other species, though. This is because scientists have never conducted or even found an animal exposed to sea star wasting syndrome besides sea stars. The sea star wasting syndrome can also happen in other locations. The disease started off in the west coast of Washington. Then it spread along the coastline from Alaska to California. Sea stars affected by the virus are also found north of Vancouver, British Columbia, and Monterey, California, as well as in Seattle, Washington, Southern California, and along the coastline from Mexico to Oregon.
First, a public survey will be conducted on where sea stars have been affected by sea star wasting syndrome. Several sea stars will be collected by hand while in the water, and tests will be run to make sure there is at least one male and one female. Once one of each gender is found, the others will be returned to the water. The sea stars that are kept will be used for gene splicing. A gene is split by certain parts into a result known as a chimera, and scientists use these products and make them into a circular shape of DNA called a plasmid. Other scientists use a complicated technique in order to clone genes from a specific organism's DNA. The sea urchin's gene will be cut out of its original DNA. The sea star's gene will also be removed from the original DNA. The genes will be combined to make a new gene, and new gene chains will be checked over to make sure that its position and composition in the DNA are not misplaced. Then, the new gene will be added to the male and female sea stars. With the new gene, the sea stars will mate and produce baby sea stars with the gene. After a while, the genetically modified sea stars will return to the water, and the sea stars already affected by the disease will die. Sea star population started off very low. However, starting in the 8th generation, the sea star population began to dramatically increase. By the 12th generation, the sea star population was higher than the category low. The sea urchin population began in the middle of low and medium. The number of sea urchins finally reached medium during the 7th generation after years of going from low to medium. For the first time, the sea urchin population was above medium during the 10th generation. The kelp population started in between medium and high. The amount of kelp was at least high, if not more, from the 3rd generation to the 10th generation. However, in the 11th generation, the kelp population dropped below high for the first time in 10 generations. The kelp population was the highest population, as the sea star nor the sea urchin populations were higher than high.
And cut!